Hello, dear students. Today we will be covering the last part of photosynthesis, that is a few experiments for the process involved in the process of photosynthesis. But before we study the experiments, we need to study one topic called destarching. This process called destarching basically means removal of starch from the leaves of a plant. A plant, especially the green leaves, produce glucose during photosynthesis. This glucose is immediately stored as starch in the leaves. At night, this glucose is translocated as sucrose. But if you keep a plant in darkness, in complete darkness for 24 to 48 hours, that means 1 to 2 days, you will be removing all the starch which was stored in the leaves. The leaves will be using that starch for process of respiration. They will convert that starch into glucose, and thereby the starch will be removed from the leaves entirely. Now, why is it required? Once the starch has been removed from the leaves and the leaves are free from starch, any starch which will now be formed in the leaves will be formed during the experimental conditions only. So this will ensure that the starch which has been formed has been formed during the experiment only. So let's start. We will be studying the following experiments today. First is the presence of starch. Then second, the chlorophyll is needed for photosynthesis. Sunlight is needed for photosynthesis. Carbon dioxide is needed for photosynthesis, and finally, oxygen is produced during photosynthesis. So let's study the experiment for presence of starch. It's called as the starch test. So first of all, a leaf is taken and dipped in boiling water. When you dip the leaf in boiling water, then you dip that leaf in methylated spirit, which is boiled over a water bath. Methylated spirit is highly inflammable, so it is not directly heated over a flame. It is kept over a water bath. Then this leaf is dipped in warm water and spread on a white tile with a few drops of iodine added to it. Why is the leaf dipped in boiling water? It is dipped to kill the cells. The living membranes of the cell will be destroyed and the chlorophyll can easily be removed. Chlorophyll is a very strong pigment. It will not allow detection of the starch test. So the chlorophyll needs to be removed. The leaf is boiled in methylated spirit over a water bath to remove any chlorophyll. Methylated spirit is a solvent for chlorophyll. So it will rapidly remove all the chlorophyll. Now, along with removal of chlorophyll, the leaf also turns brittle. So to make it soft, we dip it in warm water. And once it has been made soft, it is spread on a white tile and a few drops of iodine are added to it. Now, why on a white tile? So the color change is very clearly visible to us. If the leaf has starch, the leaf will turn blue-black. If it does not have any starch, the leaf will turn brown. So this is called as the starch test. Whatever experiment is done, the starch test is conducted in the end. So if we have to prove that sunlight is needed for photosynthesis, all these or any of these arrangements can be done. A black paper with any cutout, any shape is placed on a leaf while it is still attached to the parent plant. Or you can directly attach a black paper, which will prevent any sunlight from entering. If you have a shape cut out, the sunlight will enter through this area, but the remaining part will not have any sunlight. Or you can place a Genong's light screen. A Genong's light screen is a metallic device having a particular shape. So the metallic portion will prevent entry of sunlight, but the part exposed will allow sunlight to enter. So these will ensure that sunlight enters in certain parts of the leaves. So only the parts which are exposed to sunlight will turn blue-black. The remainder, which were not exposed to sunlight, will turn brown, indicating that sunlight is needed for photosynthesis. So how do you do the starch? Just in a similar manner as we had done. This leaf which we had taken, it was dipped in boiling water to kill the cells, boiled in methylated spirit over a water bath to remove chlorophyll, dipped in warm water to make it soft, and placed on a white tile with a few drops of iodine added to it. The part which was exposed to sunlight turns blue-black. The remainder, which was covered, turned brown. This is an experiment to demonstrate that chlorophyll is needed for photosynthesis. So any destarched plant is exposed to sunlight for an hour or so. After an hour, the leaves are taken. One of the leaf is taken and the same starch test is performed. The parts which are variously colored, not having the pigment chlorophyll, will turn brown, but the parts which are green, which have the pigment chlorophyll, will turn blue-black. So here, these will turn blue-black. This part will turn brown. These are called as variegated leaves, leaves having different coloration. So again, in this case also, a similar starch test was done. The leaf was boiled in water to kill the cells, boiled in methylated spirit over a water bath to remove chlorophyll, dipped in warm water to make it soft, and placed on a white tile with a few drops of iodine to it. And the areas which were green turned blue-black. The areas which were variously colored but not green, they turned brown, indicating that chlorophyll is needed for photosynthesis. The next experiment is that carbon dioxide is needed for photosynthesis. We have two bell jars. In one of the bell jars, we have a thistle funnel having soda lime and a pot which is covered with polythene and an intact destarched plant. We have potassium hydroxide solution in a small container placed inside. Now, why we have done this? Soda lime will prevent any carbon dioxide from entering inside the bell jar from the top. If any carbon dioxide is present in the system inside, it will be absorbed by potassium hydroxide solution. Both of them are solvents for carbon dioxide. The second bell jar is your control setup. Here we have pebbles instead of soda lime, and we have water, which has replaced potassium hydroxide solution. So there is normal atmospheric air. If both of these leaves are taken and the starch test is performed on both of them, here it will turn brown. Here the leaves will turn blue-black, indicating that photosynthesis requires carbon dioxide. Carbon dioxide was not present here, so no photosynthesis occurred in this condition. This same experiment can be done in a different manner also. And then it is called as MOLL'S half-leaf test. So what is done in MOLL'S half-leaf test? We have a leaf, half of which is inserted inside a jar having potassium hydroxide solution, and half of it is still intact, kept outside, dipped in water. Okay, you can do it with one leaf while it is still attached here to the parent plant also, or you can do it when it is separated but still dipping inside water, because we want the leaf to survive for some time. Now, the part of leaf which is inside has all the conditions. It has sunlight, it has moisture, but it does not have, it has chlorophyll also, but it does not have any carbon dioxide. But the leaf which is outside has water, has sunlight, has chlorophyll, has carbon dioxide. So when the starch test is performed on this leaf, the part of leaf which was inside will turn brown, and the part of leaf which was exposed outside will turn blue-black. So this is called as MOLL'S half-leaf test. This also shows that carbon dioxide is needed for photosynthesis. Okay, so this is how MOLL'S half-leaf test is done. In a split cork, we place half the leaf outside, half leaf inside the glass jar having potassium hydroxide solution. So this is how the leaf will turn. This part will become brown, which was inside the glass bottle, and this part which was exposed outside will turn blue-black, indicating that carbon dioxide is needed for photosynthesis. So here also we will be doing the same starch test. We will be taking that leaf, dipping it in boiling water to kill the cells, heating it in a water bath in methylated spirit to remove chlorophyll, dipping it in warm water to make it soft, and finally placing it on a white tile with a few drops of iodine. The part which was exposed outside will turn blue black. The part which was inside the glass bottle will turn brown, indicating that carbon dioxide is needed for photosynthesis. So you can see starch test is performed after all experiments, because this is the test which will tell you whether or not the particular condition is met or is required for photosynthesis. The next experiment is that oxygen is evolved during photosynthesis. This experiment is pretty simple. Any aquatic plant is taken and placed under an upside down funnel. So we place two small supports on the two sides of the funnel to ensure a rapid movement, a quick movement of water inside and outside. And we invert a test tube full of water over the stem of the funnel. And we place this entire setup in the sunlight. After some time, we notice bubbles of a gas rising. That gas is nothing but oxygen. How do we know that gas is oxygen? If a small lighted splinter is brought near the gas, once a large volume of the gas has been collected, it bursts into flame, indicating that the gas is a support combustion. We can perform this experiment using water having little sodium bicarbonate. Now, sodium bicarbonate provides more carbon dioxide. More carbon dioxide means more rate of photosynthesis. So this helps to increase the evolution of oxygen, and the oxygen produced would be made, the amount of generation of oxygen will increase in this case. So when you add sodium bicarbonate, it will increase the rate of production of oxygen. So again, we test the oxygen by bringing a glowing splinter. If the splinter bursts into flames, it indicates that the gas is nothing but oxygen. So this is how this experiment is done in the lab. We place hydrilla plant with small supports under an inverted funnel, and we invert a test tube containing water over it. The gas which collects on top is oxygen. So this tells you that oxygen is evolved during photosynthesis. So with this, we have finished the experiments on photosynthesis. Next, we have the last topic that is called as the carbon cycle. You know that green plants produce oxygen. Green plants fix carbon dioxide, and they fix this carbon dioxide means they use this carbon dioxide in the process of photosynthesis. And while using this carbon dioxide, they release oxygen. This oxygen is taken up by animals, taken up by plants themselves also during process of respiration. When animals and plants die by decomposition, this carbon content, this carbon which has been formed by the plant, the decaying of the plant matter, animal matter, all this carbon content comes back as fossil fuels or fuels which can be used for combustion process, and ultimately again carbon dioxide comes back in the atmosphere. When animals and plants respire, they also produce carbon dioxide. So this carbon dioxide is again taken in by plants. So plants again use this carbon dioxide. So in other words, that means the carbon dioxide enters the plant body, enters animals via the plant systems. And when both animal and plants die, the same carbon is returned back to the atmosphere. So this carbon cycle continues in the nature. It is maintaining the amount of carbon in the entire world. Otherwise, also, every food chain, every food web begins with a plant. So we say that green plants are the ultimate source of food and nutrition for any kind of food chain or any kind of food web, whether it is terrestrial, whether it is aquatic, whether it's a grassland, they all depend upon green plants. So in other words, we can even say that all flesh is green. That means whatever we are having, we are basically obtaining it from plants which are performing photosynthesis because of the pigment called chlorophyll, which is green in color. So whatever our kind, whatever our nature, we get energy from green plants. And all green plants are green because of chlorophyll. So in other words, it's a very common quotation. All flesh is green. So I hope you have understood it all. Thank you very much and God bless you all.